Hold on. Is this on? Yeah.
Hello, my name is Paola Ehrlich,
And I'm Kristy D'Alessandro.
and we will be presenting about BACE1 and BACE2. So to start off, we are going to give some background information on BACE1. So BACE1 is a transmembrane protein that functions as a dimer. It's mainly found in the brain and pancreas, but it is most active in the brain. The general function is to form myelin sheaths for peripheral nerve proteins. It is found on chromosome 11, and at the protein level, it's also found in mammal tissues, and it's a proteolytic enzyme with two active sites. It is found to process amyloid precursor protein, which is better known as APP, which then forms amyloid beta, as seen here. It is classified as a class 3 enzyme, which means it is a hydrolase, and its subclass is aspartic acid endopeptidase due to its breaking of peptide bonds. It has a counter protein, which is known as BACE2. BACE2 is believed to be more astrocytic, which is a subtype of glial cells in the central nervous system, whereas BACE1 is more neuronal. And in the next slide, we are going to talk about the structure of BACE1. So you can see there's So the side chains of aspartic 32 and 228 in the active site are represented by a red ball and stick model, as you can see here in figure A. And the displacement from inactive to active structures is indicated by the red arrows, which are seen right here. You can see the front view of the flap and the tyrosine 71 in structure C. This would be the flap, and then it goes from active to inactive. And then the difference in location in the vicinity of the active site, cleft and flap between the active structure, which is yellow, and the inactive structure is blue, is seen in picture D. You can also see the hydrogen bonding, probably not too well right now, through these dashed lines right here, for example, between serine 35 and aspartic acid 32, to show how strong the bonding of the enzyme is. Now we will talk about BACE2. There is not much information found on BACE2 due to the fact that there haven't done much research with the enzyme. However, it is found to be 75% homologous to BACE1s. It functions more as an alternative alpha secretase, as an antagonist of BACE21, as I said earlier, and it cleaves the APP between phenyl 19 and 20 to abolish alpha beta production. So it's located on chromosome 21, unlike BACE1, which is located on chromosome 11. It encodes integral membrane glycoprotein that functions as aspartic protease, and it cleaves the APP into amyloid beta peptide, and it's also found in the brain and kidneys of mammals. In the next slide, we're going to talk about the structure of BACE2 compared to BACE1. Both of these images would be BACE2, and then this image right here would be BACE1. The general difference in BACE2 and BACE1 would be the different conformations, which the labeled loops, which are seen here, will take, which is the difference that they'll have in their conformation, and also the difference that they will in how they will react and how they will process the APP and everything. Aside from that, there's not very much difference between BACE1 and BACE2, since they're 75% homologous. And now Kristy will talk to you about our research.
Okay, so the research we found was looking at Alzheimer's disease and the effects that BACE1 and BACE2 have on it. So this study is different from the rest because it's the first that independently measured BACE1 and BACE2 in patients with Alzheimer's disease. So first of all, the first thing you need to know is that Alzheimer's is presented with the buildup of plaques, and these plaques are made of the amyloid beta protein. Here is an image of how the amyloid beta protein is made. So this beta right here is our BACE1 enzyme. And so first you have this APP, which is your starter protein. Your BACE1 will come and cleave right below to create the APPS beta. Once this is cleaved, the gamma secretase will come and make a second cleave right at the bottom, and this will create your amyloid beta proteins that will go on to form plaques in the brain. These plaques will actually stop neural impulses in your brain, which is what leads to the degradation of your neural inhibition. And then the other part is your APP can also be cleaved by the alpha secretase, which will cleave in a different place to create an APPS alpha protein. By doing this, when gamma secretase makes its second cut, a P3 is created instead of an alpha beta, or an amyloid beta protein, which we will talk about later on for our what we think you might be able to do to help with Alzheimer's. So next we're going to look at the materials and methods. So the materials and methods, they took samples of patients' brains with Alzheimer's in them, and they collected samples from the University of Kentucky and University of California. They had samples of brain from the frontal cortex of patients without Alzheimer's and patients with Alzheimer's. Then they also had isolated kidney samples from rats with Alzheimer's. Then they took these samples, they homologized them to create the liquid with all the samples in it. Then they centrifuged it to create a pellet, and these pellets were then used to do the studies on. They used these tissues to analyze the activity of BACE1 and BACE2. So the results are here. This is when they looked at the substrate levels of BACE1 and BACE2 in the brain and the kidney. So as you can see, BACE1 substrate was very present in the brain and not present at all in the kidney. So this indicated that the activity of BACE1 was very prevalent in the brain, while BACE2 was somewhat prevalent, but it was much more prevalent in the kidneys. Our second result is the Western blot from their trials, and this was just to see if the BACE1 and BACE2 were in relatively same amounts within the sample. So on the left you can see BACE1 and BACE2 is on the right. These first couple are the control, followed by the Alzheimer's. In BACE2, they did the mouse kidney. They did not perform the mouse kidney on BACE1 because it was not prevalent in the kidneys at all. Then you're followed by the controls and then the Alzheimer's. Next is the results for
ELISA.
For the ELISA, and you can see the star indicates that the results were significant. BACE1 is much higher. This lighter color box is the control, while the dark is Alzheimer's. I'm not sure if you guys can see it throughout the entire graph. So the results were significant for BACE1 in both the brain and the kidney samples, while BACE2 did not have significant results. This indicates that BACE1 and 2 is still present in all the samples. The last result is the control and the Alzheimer's. So the RIPA is the insoluble, the samples with insoluble detergents, while the FA is the sample with soluble detergents. This was done, and they observed a positive correlation between the amount of BACE1 and alpha amyloid beta present in the brain. So this is indicating that they had a positive correlation with the amyloid beta leading to Alzheimer's. So for our predictions, there's a lot of studies being done on inhibiting the BACE1 gene and BACE1 protein to decrease Alzheimer's. The problem with that is, first of all, Alzheimer's is a growing problem where it starts out with a little bit of plaque buildup and it gets much worse over time. So it builds on, and most Alzheimer's patients are diagnosed with Alzheimer's much later than
what they would like.
to be able to stop the treating. The other problem was there was a study done by Steve M. Harrison. He did it on mice where he completed a knockout. So he knocked out the BACE1 gene. It was a double knockout study. And he observed their behavior and noted many behavioral and physiological differences in the mice. So they have not done this on humans because they are not sure what these side effects would be. So one of our prediction is to increase the alpha secretase enzyme, because this will reduce the amyloid beta that is produced by producing more of the P3. The only problem is that we are not sure what the side effects of an overproduction of P3 would be, and we were not able to find much research on the overproduction of P3.
Here are all of our references. We really hope that you enjoyed this video and found a lot of useful information about BACE1 and BACE2. We wish we could have found more information on BACE2, but unfortunately there haven't been as many studies that we would have liked. So hopefully in the future there will be more information so that we can do a better comparison between BACE1 and BACE2. If you have any questions or comments, you can put them under our video. And thanks for watching.